Hello, I'm Alex. I'm a PhD student at the University of Glasgow, and today I'm going to talk about how to use topic modeling to detect cellular crosstalk in single-cell RNA-seq. Over the past few years, single-cell RNA-seq has been gaining extreme popularity, and it's been used to study heterogeneity of cell populations as well as to analyze processes through the use of pseudotime and trajectory inference. Cell-to-cell interactions are vital for numerous biological processes, from differentiation and development to disease progression. Currently, there are two main strategies for studying interactions. One relies on ligands and receptors. This strategy uses curated resources of ligands and receptors, and as such is limited to known biology. Furthermore, curated databases predominantly exist for human data and mouse orthologs. An alternative to the ligand receptor-based strategy relies on having access to physically interacting cells. Earlier last year, a paper was published that describes one such approach, known as PIC-seq, or physically interacting cells sequencing. On figure 1, we've shown how the PIC-seq approach operates. Following tissue dissociation, cells are stained for mutually exclusive markers. Then, the single and the double positive populations are sorted. On the computational side of things, the single positive populations are clustered, and then synthetic reference profiles are built. By comparing the synthetic reference profiles and the double positive populations, genes that change their expression as a result of interactions are identified. Here, we present a novel method that does not require prior clustering or generation of synthetic reference profiles. Our method is based on latent Dirichlet allocation. In our formulations, cells are equivalent to documents, genes are equivalent to words, and topics are groups of covariant genes. We begin by fitting our LDA on a reference population. That reference population can be sorted cells or cells before interaction has occurred. Once we obtain cell by topics and genes by topic probabilities, we fix some of the topics before we fit a second LDA on the interacting population. It is the result of our second LDA we use to rank genes that change their expression as a result of interaction. We test our approach on a range of real and synthetic datasets. However, here we focus on two of the real datasets we've analyzed. The first dataset we use in our analysis has been generated by the PIC-seq approach described earlier. This is a dataset of T cells and dendritic cells taken across different time points. The first LDA we fit on two separate co-cultures of T cells and dendritic cells. On figure 3a, we can see that we identify topics specific to one cell type over the other. For example, topics 12 and 23. On figure 2b, we've shown some genes that rank amongst the top ones for those two topics, and they are specific to one cell type over the other. Next, we fix the topics from the first LDA and fit a second LDA on the interacting population of T cells and dendritic cells. Here, on figure 3c, we present some of the genes we identified to be changing as a result of interaction based on our ranking approach. Our top candidates for interaction include genes related to immune response and cell adhesion, another evidence of the physical interaction between the cells. Next, we analyze a bone marrow dataset. Our reference population includes sorted bone marrow cells where our interaction population is based on dissociated doublets. We follow the same analysis procedure described earlier. Amongst the top-ranking genes, once again, we see adhesion molecules as an evidence of interaction. Some of the genes we've identified from our analysis can be seen in figure 4. And to summarize, here we propose a method that does not require curated resources of ligands and receptors, prior clustering, or generation of synthetic reference profiles that also allows us to identify genes that change as a result of interaction. This is the first use, to our knowledge, of LDA that detect genes that change as a result of interaction. We produce a comprehensive ranking of candidates of interaction comparable with the original publications of the discussed datasets, as well as identifying some further genes that might be of interest for further exploration. For any questions or further information, don't hesitate to stop by my poster and chat to me.